Welcome to this episode of Eureka. We are going to have a very interesting discussion with Dr. Mourya, Director of National Institute of Virology in Pune, a constant unit of Indian Council for Medical Research. Before we continue this discussion, let's take a quick look at his brief profile.
Dr. D T Mourya is presently the Director of the National Institute of Virology in Pune. He specializes in the area of microbiology and immunology and worked extensively on vector potential of mosquitoes to arboviruses of public health importance in India. He also significantly contributed in vector biology aspects of vectors of dengue, chikungunya and Japanese encephalitis viruses. Dr. Mourya did his MSc and PhD from University of Indore and has received several honours, including the first Dr. T Ramachandra Rao ICMR Award in the year 1990 for his work in the field of medical entomology.
Thank you for giving us this time and permitting us in this lab, Microbial Containment Complex. This complex is one of your achievement. You have built it in some sense brick by brick. You have put it together, you have designed, you have ensured that this place is a world-class facility for this institute. Can you tell us something about this place?
In fact, the vision to create this facility was from our earlier director, Dr. Gupta, 20 years back.
20 years back. Oh.
That time this land was purchased, but somehow this complex came up after 2001. After 2000, these labs came up and number of state-of-art facilities have come up in this complex. We have 2 BSL-3 laboratory and 1 BSL-4 laboratory, which is countries.
BSL means Biosafety Lab.
Biosafety level.
Yeah, Biosafety level. Oh.
So 1 BSL-4 lab, and that is the so far unique and only lab in whole Asia, which works in health sector.
What does this facility provide? I mean, what kind of work you can do here, which you cannot do, let's say, in other places?
This facility was needed very much, and this requirement was felt since a long time. The reason is, when we see that newly emerging infections which are highly, or we call now as high risk consequences, the moment anybody is exposed to those infections, the probability of death is extremely high.
Unlike a common cold.
Unlike common cold, where a person doesn't succumb to the infection. Now, in these kind of infections, we have to see that the scientists and workers, those who work in the lab, their life is secured. And that's the reason this biosafety science and biosafety level, higher level labs have come up. And that was the reason we did. And we have been working on number of viruses which are high risk consequences. Besides that, this lab is also required for carrying out research and developing reagents for this kind of infections when they have arrived here. When we develop reagents, it means that larger volume we have to do. For that, we need higher scale up facility. And when we do research and we are trying to find out solutions for that, or recombinant DNA technology when we are using. So, we have to have a very secure place to see that any emerging virus shouldn't go out in the environment. So, that's why this
facility has been created.
Yes, yes.
One of your strength, in fact, you are known world over for that particular strength, which is developing biosafety level laboratories. And you have written a manual which has been published by this institute. So, what is this biosafety level? I mean, how do you categorize? How many levels are there?
There are 4 biosafety levels, and in fact, these levels are defined. Those science students, if they look at that, whichever branch they come, whether it is botany, zoology, microbiology, they see the classification in phylum, class, order like that. But for us, all the microbes on the earth are classified into 4 groups.
Okay.
And they are called as risk group 1, where absolutely there is no risk to the humans and even to the plants and other.
Example would be?
Example, say E. coli.
E. coli. Okay.
Normal E. coli infection, even the students in junior college also they work on that. So, they can work. So, those kind of laboratories where they can handle this is biosafety level 1. But majority of the infections which you read across in the newspaper every day, 99% infections are in risk group 2.
Risk group 2.
So, you require biosafety level 2 laboratory for that.
One of the example of such
Say dengue, chikungunya, Japanese encephalitis like that, hepatitis.
they will be in biosafety level 2.
Right now, you are sitting in a biosafety level 2 laboratory.
Okay.
If you see anywhere in the country, any city, every third street you will have a pathology lab.
Yeah.
They are all biosafety level 2 laboratories.
By
they deal with this, they provide diagnosis,
Okay.
that urine sample, blood samples, throat swabs, etc. Biosafety level 3, where those agents we handle which cause threat to the individual,
Okay.
not to the community. The scientist working and get infected with that, that's a life risk for his or her life. And risk group 4, biosafety level 4, right now in this complex where you are sitting, we have a BSL 4 laboratory here. These are high consequences viral infections, where probability of succumbing to infection is extremely high.
Examples like?
Say example is Crimean-Congo hemorrhagic fever, which is present in our country. Recently, people have heard about Ebola threat worldwide. Everybody was worried about that. Marburg's, many infections like that. They belong to risk group 4. Risk group 3, I was giving example, Cassonero forest disease, is though it is endemic in our country, otherwise for WHO and CDC it is considered as risk group 4 in other countries. So, this is how the risk groups are
these are all various biosafety levels. And then your manual talks about how to handle such
to handle these organisms in different level of laboratories.
Another area of your strength, I mean, if one looks at your scientific career, has been your work with regard to viruses which are transmitted by arthropods, like malaria, chikungunya. In fact, your papers, chikungunya, written in 1980s, kind of predicted that we should be aware of it. Yes. Can you tell us about your work on this area?
Well, when I joined this institute in 1977, since then I have been working on arboviruses, and this last 4-5 years in my career, if you look at that, still I am concentrating more on these emerging viruses. Majority of them are arthropod borne.
Okay.
Even Crimean-Congo example I said is arthropod virus. So, your question was that how I got into that. Initially when I joined this institute, the mandate of this institute was to work on arboviruses only.
Okay.
So, in late 80s, this became a National Institute of Virology, and then we started working on various different groups of viruses and different specialties we established later on. So, since then I have been working on different arboviruses.
Okay. Your work on chikungunya, which I think spans more than 3 decades now.
Yes.
Yeah. So, can you tell us what was your work? What were you trying to focus on? What were your research questions?
My research question those days in say late 70s and 80s, that time it was said and papers were published, and it was said that this chikungunya has disappeared.
Okay.
As a scientist, I did not believe, and my hypothesis was that this is, though viruses are obligatory parasites and without living organisms they cannot survive, and chikungunya virus transferable transmission is also not well known. So, how the virus survives, and this is what the prediction from the other scientists, that it has disappeared. And my suspicion was that we are not actually keeping the diagnostic test in our arsenal and checking it, testing in the people that whether this disease is there or disappeared. So, a lot of work I focused on that. I worked on the natural cycle, that where it can occur, there may be any occult cycle of chikungunya in nature where it survives, or any other animals involved in that, how the vectors transmit, how much time intrinsic factors it is there, what are the factors affect the transmission of chikungunya, which strain of chikungunya virus is very efficient, or which strain of vector is very efficient, and those combinations, if it come, there may be massive outbreaks. So, these are all research questions which I was working with.
In some sense, you sort of predicted that chikungunya is there in the shadows.
And then I proved it in
and
2001.
Unfortunately, it appears to be true today, where we are seeing the resurgence of chikungunya and malaria and this kind of infection. We will take a very short break. We have been having a very interesting question. After the break, I am going to ask him, what is his favorite virus? Don't go away. After the break, we will continue this conversation. Welcome back to this episode of Eureka, and we are having a very wonderful conversation with Dr. Mourya, Director of National Institute of Virology in Pune, a constituent unit of Indian Council for Medical Research. Who was, if I can ask, was your role model?
I joined in 77. That time my immediate boss, Dr. Thanda, I consider as the role model, and I was very young guy that time I had joined. I was completing my PhD, and I learned a lot from him, and I was very closely associated as research assistant to him that time, and a long association with him, really made me look into
why do you think him as a very important person who kind of shaped your career, life in a very major way? What kind of characters?
What I liked him is he was a great thinker, and he made me think all the time. Though even when a scientific question is coming up, he knew the solution. He used to give me a challenge. Think about it. No, you are still not right. Think about it. Go and read in the library. Find this. Do this experiment. And this is how I learn things. How to proceed in research field. And he was very supportive, very encouraging person, and a lot of knowledge I gained from working with him.
I told our viewers that I am going to ask you this question. What's your favorite virus?
Of late, if you say Crimean-Congo hemorrhagic fever, is very, in short, it is called as CCHF.
CCHF.
Yes. And this virus we discovered recently in 2011. First time in India, and this also belongs to very high risk group, risk group 4. And your question was why I am interested in that, because the moment we identified this virus and we investigated outbreaks, various outbreaks when they are occurring, and our initial work showed that when the person gets infected, basically the infection arises from the rural areas, and when the patient reaches to the hospital, there cases occur in the hospitals where doctors and nurses have died.
Oh, okay.
And a number of episodes have occurred of that kind, and sometimes contacts also died. So made me really, interest came up that what best we can do to save the lives of people. When we worked on it, and of course now we know a lot about it. In the last 4-5 years, this institution and my team has done a lot of work on it. We worked very closely with Gujarat government, with health officials, and explained them also that how we can reduce this risk, how we can quickly diagnose what are the things, and several publications also will come out on that. But still, as I said, that why I am interested, because we could really do in public health, whatever work we do, we don't have figures to say that this we did, so we saved so many lives. Okay. In healthcare system, very easily you give the figures, how many people you treated and saved. But a lot of work on CCHF we have done, and still a lot of research work is needed in the country on that. I still feel that we should produce vaccine on this virus to save lives.
Yeah. Very interesting. I mean, there are people who catch butterflies as their hobby. There are people who collect, let's say, old coins or stamps as hobby. And I'm told you are one person who goes around, and your friends and colleagues in this institute go around the country looking for viruses which can cause diseases. Can you tell us how do you do that? I mean, where do you go? How do you find, what do you do to trace these viruses?
This scientifically speaking is called as hunting for viruses.
Hunting for viruses.
Yes. Now, hunting for viruses is not a simple example like catching butterflies, but there also science is involved. If there are specific species they want, they have to go to a very specific place. The same is the case here. Now, when we are hunting for viruses which may emerge or which are emerging from bats, then we go to the areas where bats roosting is there, the colonies are there. So, we go, we trap them, we collect the samples from them, bring those samples in liquid nitrogen, we try to isolate virus from there if at all anything is there, and we work on that. Similarly, when we talk of influenza, say bird influenza, so the domestic and wild birds which are coming there, so we go to those places where they are there and collect the samples, the droppings and environmental samples like water samples collected from those areas, and we test that those samples to see that if any other viruses they are carrying, because the migratory birds come from different country. So, like that some other viruses if you are hunting which are rodent borne, so then we go to those areas if they are needed. We are hunting in the domestic rodents, in the villages across, otherwise in the forest areas we collect the rodents. So, like this specific viruses which we are interested in, it is also called as proactive public health research. And we do get of course indications when we see that this kind of viruses are that group of viruses which are emerging from other continent or other countries. So, this is how we go for hunting of viruses.
Interesting. You come from a not so privileged background, and you had a struggle to come to this position, and now you are heading a very important institution in this country. What was your story?
My story is like many students in this country, they are facing whether yes, economy is very important, getting support from the parents for higher educations and going ahead is important. And sometimes students may be very brilliant, but they are coming from such a place or maybe from a village background, may not get opportunity to come up. So, there are many factors like that, and but yes, hardship was there. I have only one message I can say to all youngsters: if you work hard and you are really dedicated, there is always said no position is vacant on the top.
If you are dedicated, the position is vacant at the top. That's an important message that Dr. Mourya, Director of National Institute of Virology, is giving to all of us. We'll take a very short break. We'll continue this conversation. We want to ask him what this institute's major contribution has been in last 50 odd years of its existence. After the break, keep watching Eureka. Welcome to this episode of Eureka, and we are having an interesting conversation with Dr. Mourya, Director of National Institute of Virology, a constituent unit of Indian Council for Medical Research. This institute was founded in 1953, so almost about 60 years it has been in existence. Suppose if somebody wants to ask you what this institute has done for the nation building in the last 50-60 years, what would be your top three?
I would say the arboviruses. Right now, if you pick up any newspaper, you see there are 2-3 diseases of public health importance. So, I will give examples of those first. Is say dengue, chikungunya, Japanese encephalitis and influenza viruses. So, these are the all 4 some or other way. Now, this institute in 50s discovered first time the presence of these viruses. Say
this
dengue,
institute
chikungunya.
discovered the presence of these viruses 50 years ago in India.
Yes, in India, and then the basic scientific foundations were made by this institute. That what are the vectors, where these vectors are found, where they breed, what can be done for prevention of these infections, what is the incubation period, what exactly happens when the outbreaks occurs, and what kind of syndromic, we follow the syndromic approach, but what kind of variations we see in the patients, what is the best sample for providing the right diagnosis, and what time the sample should be drawn. Say for example, if we take example of dengue, so this is what we showed long time back, that in acute cases when the person comes down with this disease and fever, so first 4-5 days molecular tests are really good. That time IgM, the antibodies don't develop, and if those tests are done before 5 days or 6 days, then test will be negative, but the person may be positive for that. So like that, there are many basic research questions were answered by the institution. The institution also provided many diagnostics, tests, kits, innovations for that, how to for the diagnosis of different diseases here, and great contribution, as you asked, that I would say that the natural cycle of Japanese encephalitis in our country and natural cycle of cassinute forest disease, where the virus was discovered and up to the killed vaccine was produced and developed, and it was passed on to, technology was passed on to that level of. So these are all great contributions of the institute. These are public health important viruses which we are talking, but the institution has been responsible, has done a lot of work in hepatitis viruses, because there again very basic information what was needed initially, that was provided, and which really helped in saving lives, particularly in, if we talk of hepatitis E in pregnant ladies, hepatitis A outbreaks, and how to save, and hundreds of people come down with the infections. And influenza viruses, you have heard of pandemic influenza. So since 2006, a lot of contribution this institution has given. The important thing what the institution really made, and we developed various network centers and provided support. The contribution, if I put it, the first thing is enhancing the lab capabilities of several labs so that they can provide right diagnosis in time. We also worked on it, on these viruses which are emerging, and understand their full genome, isolate them, and see that if there is any drug resistance coming up in that, if it is that, so that interventions are needed. State can be informed about that, and then it can be contained on that. There are various areas institution has worked on it.
So friends, when one of your relatives is suffering from chikungunya and the person goes to a hospital and the person is being treated, perhaps what you see is that hospital and the doctors and that technicians over there, but then there is a hidden hand of National Institute of Virology behind all this. I am not exaggerating, right? Yes. Okay. What we talked about now is the past 50 years. The institute was started in 53. We are in 2016. We have entered 21st century. Do you think there is a need to reinvent the institute? What kind of new challenges we need to take on and focus?
I agree with you. There are new challenges are there. New challenges in term, I will put it two ways, because the science and technology go on. So new and new inventions are helping us to go more and more closer and understand these emerging infections. And the second side is that newly emerging infections, whether mutations getting accumulated or naturally viruses are getting mutated and coming. Zoonotic infections are really more important now. These newly emerging infections mostly are from the animals. So the zoonotic infections are coming up. So these two areas really institute has to think in more. And new, as I said, that technology is moving faster. New technologies are getting added. So we need to, the old what we used to do in 50s, 60s, 70s, now scenario has changed. We are switched on now more on molecular systems. So like that, as science is moving, the institute is also rolling in those directions.
Moving to this new directions, taking on the new challenges.
Yes.
So on behalf of our audience and this country, we thank you for being forthrightful and making this innovation as a director of this institute. To continue our conversation, let me move to another area. We talked about the strength, we talked about the potentials. What do you think is a weakness in this area in this country? What do you think is a major weakness that we should address so that we can face tomorrow's challenges?
Major weakness, I would put it in a term that whenever there is medical emergencies, we feel that the good capacity labs are lacking in our country. And second thing what we really miss is a very strong surveillance system. Now
good laboratory
surveillance system. Now surveillance system will be sustained by the good laboratories. So what is needed is enhancing the lab capabilities, not only in national institutions, but in across the country. Because country has very wide topographical and climatological situations in our country.
So you need labs in different
regions in all the places, good labs.
Good labs.
And so once they are connected, they will provide support in surveillance.
See, one of the common misperception among the public is that India is dirty. That's why we have these diseases. But as a scientist, do you agree that?
No, I don't agree. Of course, to some extent you can say that hygiene, personal hygiene, if we talk in people in general, is definitely awareness is needed more and more. And we see that now our national programs are also taking the whole country ahead on those parts. And if you succeed in that, many of the diseases will go. Many of fecal-oral route infections, dysentery, diarrheas and all, they will all reduce as when the cleanliness will increase. But I would give you example that you can't just say that India at that stage. If you go across around other adjoining countries in this region, you will find that India is doing much better.
Yeah. That's because of the strong public health system and research system in India.
Science, technology, education, in all respects, India is doing very good.
Thank you. Thank you for being with us in this conversation. It has been a very wonderful conversation and we learned quite a lot.
Thank you.
Thank you for watching this episode of Eureka. Continue watching Eureka. Next week, same time, we'll meet with another conversation with another scientist.